Hello, welcome students. We shall discuss today about the prokaryotic cells. We know that on the basis of nucleus, the cells are divided into two. One is prokaryotics and next one is eukaryotics. You know that pro means primitive, that is primitive nucleus, means the nucleus is not surrounded by nuclear membrane, means that type of nucleus which is ill developed is also called incipient nucleus. So this nucleus is also known as incipient nucleus. Its example is bacteria, blue green alga. In the blue green alga, you will get the example of nostoc, anabena, mycoplasma, PPLO, or mycoplasma PPLO, that is pleural pneumonia like organism. These are the examples of prokaryotic cells, and you know mycoplasma is having no plasma membrane, and these are the smallest cells. Now we shall see about the bacteria. Singular bacterium. You all know about the bacteria, understand? Its singular is bacterium. It is of someone is round in shape, that is called coccus. Someone rod shaped, that is called bacillus, rod shaped. Someone comma shaped, that is vibrio. Someone spiraling shaped. These are the following shapes of the bacteria. Now we shall discuss the bacterial cell. Now we shall discuss the bacterial cell. We take a cell It is oval shaped arterial cell, and this is called plasma membrane. Understand? And above the plasma membrane there is another lining is also found. Another lining is also found, that is called cell wall. That is called cell wall. It is just above the plasma membrane. It is made of peptidoglycan. It is made of peptidoglycan, and in some cases above the cell wall and another coating is also found. That is known as capsule. This is the capsule. In some cases it is found in some bacteria. It is also some bacteria contain capsules, some do not. Understand? Now second, glycocalyx. It is made up of glycocalyx. These are the surroundings of the bacterial cell. First one is plasma membrane. Above that the cell wall made of peptidoglycan, and above that the capsule is found. Capsule in some cases. Inside the protoplasm there is fluid contents. This is called cytoplasm. This is called cytoplasm. In that, and you know that in the prokaryotes membrane bound organelles like mitochondria, Golgi bodies, endoplasmic reticulum, absent. These are found in eukaryotes, but in that in the prokaryotic cells these organelles are absent, not found. Understand? But there is something which ribosome is found in both. This is the ribosome. It has 7ts type, which is made up of two parts. One contain 50s and next to contain 30s. Once in total that is 70 years. This fashion is like that. So 50 years and this is 30 years and total 70 years. So this is, and the main thing is that in the cytoplasm of the prokaryotes, some on the outer side of that some finger-like projection is found. These are called fimbri. These are called fimbri. It helps into adhesion. It helps into एडहेशन यानी चिपकने में, किसी सबस्टेशन से चिपकने में ये हेल्प करता है. सो एंड दिस आर नोन एज फिम्ब्री. Except this, there are some foldings are found in that. These are called mesosome. These are called mesosome. And this mesosome is on respiration. It means in that it acts as mitochondria of eukaryotes. It takes part in the respiration as the mitochondria do in eukaryotes. Now that in that the circular DNA is also found. This is and some DNA is found in the circle in the center, and that is in the non-division state, and at the time of division they comes to the mesosome. That is the DNA, circular DNA found in the center, and sometime at the time of cell division it comes to the mesosome, because mesosome also plays in its division and sometimes this DNA found in a very small forms, that is known as plasmid. That is known as plasmid. Plasmid is also found. That is a short DNA, and this plasmid contain 3 type of gene. One is F gene, second one is R gene, third one is cold gene. So these are the genes which action just here. If the plasmid is present, that will be, the bacteria will be positive. Plasmid is present, that will be positive, and that means that is donor. If the plasmid absent, that is negative, and that will be acceptor. So this is the due to presence of plasmid. If the plasmid is present, there will be a somewhat long projection is also found, that is known as sex pila. That is known as sex pila. Only difference is fembril, it is somewhat short, and that will be long. Now this plasmid contain three type of genes. F gene, that is fertile gene, understood? R gene, that is antibiotics for resistance, antibiotics for resistance, and this call gene is also, it is also resistance for such gene, for such chemicals which excreted from the E. coli. E. coli say, E. coli is one of the main bacteria which is found, that is, and on which many experiments has done. E. coli, that is Escherichia coli, understand? So these two are the resistance unit genes, and these are the fertile genes. So now this is the sex pila, and this is the nature of the cell, a bacterial cell. And one thing you also get, some bacteria is gram positive and some one is gram negative. Understand? These are the on the type of on the base of staining, that may be gram positive or negative, and because this name was given, that is done by a scientist called, his name was Gram. So on this gram positive and gram negative, if the bacteria is undergoes gram stain, means that is undergoes staining with safranin, understands? Some bacteria will got purple color, some will not. Understand? Who will got purple color, that will called positive, that who does not, that is called negative. Means actually the bacteria, this is the bacteria, it contains a coating called peptidoglycan. Understand? When that is stained by gram stain, the purple color will present after washing. But in some bacteria, this is peptidoglycan layer, and above this There is also lipoprotein coating. Lipoprotein does not allow the stain to go inside and to react with the peptidoglycan. So it at first it takes color, and when it washes, the colors washed away. So it has no colors. नो यानी कहना है मतलब कि जो बैक्टीरिया के आउटसाइड में केवल पेप्टिडोग्लाइकेन की होती है, कोटिंग होती है, उस पे स्टेनिंग चिपक जाता है और उसका वो वॉश करने के बाद भी नहीं साफ होता है, समझे, इसलिए वो पर्पल कलर एज जो करते हो वो बच जाता है। लेकिन जिनमें इस पेप्टिडोग्लाइकेन के आउटसाइड में एक एक्स्ट्रा था लाइपोप्रोटीन की होती है, वह लाइपोप्रोटीन इस स्टेन को पेप्टिडोग्लाइकेन तक नहीं जाने देता है, जिससे वह कुछ समय के लिए कलर लेता है, लेकिन जो ही जब उसको वॉश करते हैं, धोते हैं, तो दैट इज वॉश्ड अवे, समझे। सो एंड बैट बिकम्स कलरलेस. So it is gram negative and this is gram positive. In that positive examples, streptococci, staphylococci, and in that gram negative E. coli, salmonella, like that. So these are the some features of the bacterial cell, which is the example of prokaryotic cells. In the next video, we shall discuss the eukaryotic cells.